Okay, so site-directed mutagenesis. This is a fairly old technique and uses the standard tools of a molecular biologist, that is plasmids, primers, DNA polymerase, dNTPs and DNA ligase. Essentially, what happens is the DNA into which you wish to introduce a mutation is cloned into a plasmid, or the plasmid that already contains the DNA is selected from a library. The double-stranded DNA of the plasmid is made single-stranded and a primer is introduced that binds to the region you wish to mutate. Now the primer, just as with the PCR primers, has been specifically designed to be around 20 bases or so long and to be an exact match to the region of the plasmid DNA you wish to mutate, with one exception, the base you wish to change. And that base will be the new base, the mutation. Once the primer has annealed to the single-stranded plasmid DNA, DNA polymerase and dNTPs will be added so the remaining single strand can be made double-stranded. And finally, DNA ligase will be added to seal the nick. The plasmid is then introduced into bacteria and the DNA repair mechanisms of the bacteria will repair the mismatched DNA. The repair will result in some of the plasmids being converted back to the original sequence and some being converted to the new, therefore introducing the mutation. So by using this approach, it is possible to change a single base to explore how proteins work and genes are regulated. However, this type of work would now be carried out using CRISPR.